Hello everyone, welcome to Dr. K. Primer video lecture series presenting by Dr. K. Prem, that's me. Today we'll talk about cell cycle checkpoints. What is cell cycle basically? Cell cycle is nothing but alternating of growth and division of a cell is called as cell cycle. In cell cycle, there are two things will happen. One is the cell grows and followed by the division. This process is continuous under regulated conditions. So a cell which can grow and divide, that process is called as a cell cycle. And a cell cycle, we'll talk about checkpoints. Checkpoints. As you all know that cell cycle corresponds the growth and division, but the growth happens in the interphase and the division happens in the M phase. The interphase is divided into G1, S phase and G2, and M phase, that is nothing but mitotic phase. The mitotic phase M phase have prophase, metaphase, anaphase, and telophase, and followed by the cytokinesis, which ends the division. Then cells enter into G0. Whenever cell which is in the resting period or quiescent period receives the growth signal or a growth factor stimulation, the cell enters into G1. In the G1 cell grows. Basically, cell when divided, it is in a small size. It grows, means copying of cellular structures happens. Then once these cells grown up and they enter into the second phase, that is synthesis phase, where DNA replication takes place. Entire genome is duplicated. 2n becomes 4n. Then cell enters into G2, where again here also cell grows. Then finally it enters into the M phase, where it undergoes the various stages of the mitosis and cells divide. This is the overview of cell cycle. Then coming to the cell cycle checkpoints. So cell grows from G1 to G2, then enters the M phase for the division. But meanwhile, cell can undergo various stress conditions. During the stress conditions, its genome or its cellular components may be damaged or degraded. So before going into the next stage, every stage, the cell itself checks all its parts, whether they are in order or not. So to check this normality of the cells, there are certain points which checks these overall fitness of the cell. They are called as a cell checkpoints, cell cycle checkpoints. You can see definition. Cell cycle checkpoints prevent the entry into next phase of the cell cycle until the events of a previous phase have been completed. So every phase in interphase have a defined functions. So unless and until those defined activities are completed, then only it will move from one phase to other phase. If not, if they don't finish that part, the cell will not enter into the next phase, where they'll arrested or undergoes the apoptosis, depends on the activities. In other way also, you can say, cell cycle checkpoints allows the passage of ordered cells and prevent the entry of disordered cells and arrested. Whichever cell has completed its activities corresponds to the phases, those cells enters from one phase to other phase. If they cannot complete it because of various reasons, and those cells are stopped, are prevented to enter from one phase to other phase. So these things are done by the cell cycle checkpoints. There are 5 cell checkpoints are there in the cell cycle, which checks the fitness of the cell and various other parameters to entertain from passage of cell from one phase to other phase. If you take G1, in the G1, whenever after division of the after M phase, after mitosis, cells which are divided into daughter cells, they are G0. Whenever there is a stimulation from in the form of growth factors, then G0 to G1 start. The G0 cell enters into the G1. But during the G1, the cell size has to be increased, means cell has to grow. And to grow the cell, it requires the nutrients. It requires the nutrients. If there are enough nutrients, then only cell grows. And also it requires the growth factors. And also, if there are any DNA damage in the genome of cell, then that also repaired. If any one of them is not fulfilled during the G1 phase, then there are cell cycle inhibitor which acts on the cell cycle regulators and inhibit the progression of cell from G1 to S, and they arrest the cell to enter into the G0 stage. You see, for example, here you have this is a budding yeast. When it undergoes the division, you have 2 asymmetric cells. One is a mother cell, which is in a bigger size, and the daughter cell is small size. The smaller daughter cell has to grow in size to enter from G1 to S phase. Until and unless the daughter cells grows the size of the mother cell, then only that budding yeast cell moves from G1 to S. If the cell remains in the same size of the daughter cell, it won't entertain the movement of cell from G1 to S. Here, there are the progression of cell from G1 to S is made by the cyclin-dependent kinases. So the cyclin-dependent kinases are blocked or inhibited by the cell cycle inhibitors. You see, the cell cycle inhibitors, they are called as P15, P16, P18, and P19. They can bind to the cyclin-dependent kinase, that's a 4 and 6, and it will not allow the phosphorylation of the PRB protein, which is required for the release of elongation factor 2, that's required for the synthesis of cyclins and other proteins required for S phase. So cell cycle inhibitors such as P15, P16, P18, and P19, they'll bind to the CDK4 and 6 and does not allow to function as a kinase. Then the cell enters from G1 to G0. G1 to G0 is nothing but cell arrest. If there is a DNA damage, the DNA has to be repaired, then only it moves. So any one of these are not available, then cell goes from G1 to G0. The next point is that the second checkpoint is also called as a restriction point. Restriction point in case of mammals. In case of this is called as a start point in the yeast. So here, once suppose for example, G1 is all is well, then it has to cross the G1 to S. But it is a very critical point, hence it is called as a restriction point. Once the cell passes from G1 to S through the restriction point, then there is no requirement of growth factor. So that's why it is called as a restriction point. Here, again, at this restriction point also, cells once again scrutinize the integrity of its DNA. If it is satisfied its integrity, then only it moves from G1 to S phase through the restriction point. For this movement from G1 to S through the restriction point is mediated by the CDK2 and cyclin E. This is a regulator molecule which passes the cell from G1 to S through the restriction point. If there is a DNA damage, again the same cell cycle inhibitors P21, P27, and P53. These are they binds to this complex CDK2 and cyclin E and reduces the cyclin E concentration. So once they block the activity of cyclin E and the CDK2, again cell is arrested. Again, the cell goes through whatever activities it has to perform before the synthesis of DNA, that will be done. Then only once they finish the DNA, once the DNA damage is repaired or copying of cell structure is over, then synthesis of various proteins required for the DNA synthesis are prepared. Then that cell enters from G1 to S phase. S phase is nothing but synthesis phase. Synthesis of what? Genome synthesis. So duplication of DNA happens during the S phase. If during the replication, every part of genome has to be replicated, and there is no, and also during the replication, if at all it incorporates any kind of wrong nucleotide, and they have to be rectified. Two things: every part of genome has to be replicated, and whatever errors are incorporated during the replication, they have to be fixed. If that is the case, you see, as I told you that unreplicated DNA is there, it has to be C, and damaged DNA, it has to be rectified. These two S checkpoint checks the replication of every part of the genome and repairing of damaged DNA which has happened during the replication of DNA. So S checkpoint checks these two things. If at all if it passes the two criteria, then cell moves from S phase to G2. But if the damaged DNA is not rectified, or every part of genome is not duplicated, then the cell cycle inhibitors called as a P21 and P27 and P53, they'll bind to the S phase cell cycle regulator, that is a CDK2 and cyclin A, and inhibits the cell movement from S phase to G2 and enters into that phase. Cell is arrested, means delayed the movement of cell from S phase to G2. These are the P21 and P27 and P53 molecule are activated due to the damaged DNA, and they'll interact with the CDK2 and cyclin A and stop the movement of S phase cell from S phase to G2. And if at all everything is okay, then it goes to the G2. Then in the G2 also, again cell size is monitored. Cell size is monitored and DNA damage is monitored. So here the movement of cell from G2 to M phase is coordinated by the master regulator that is called as CDK1 and cyclin D. So you all know that CDK1 have three phosphorylation sites. One is at the 160, and 160 309 and 14 309 and 15 tyrosine. 309 160 is a activation phosphorylation, and 309 14 and tyrosine 15 are the inhibitory phosphorylations. So the CDK2, CDK1 with the cyclin B at 3 sites they are phosphorylated. If at all there is no DNA damage, the phosphatase called as CDK1 and cyclin cyclin B is dephosphorylated by the CDC25 when there is no DNA damage. If there is a DNA damage, here only cell cycle is stopped at the G2 only, right? If there is no DNA damage, then if there is a DNA damage, the dephosphorylation of CDK1 is not happens. Only whenever there is a CDK, whenever there is no DNA damage, then only CDK1 inhibitory phosphorylations are dephosphorylated by the phosphatase called as a CDC25, then allows the cell from G2 to M phase. Once the cell enters into the M phase, the most important thing in M phase is that duplicated chromosomes are distributed equally to the daughter cells. That has to happen. So when it will happen, whenever all the duplicated chromosomes are aligned in the center of the cell, that is called as metaplate, then these chromosomes will bind to the mitotic spindle and share the chromosomes equally. If the duplicated chromosomes, if they cannot sit or align in the center of the plate, then the cell will be arrested at that point. That's called as a spindle. This is called as you see, this is attachment of chromosomes to spindle or alignment of chromosomes on metaphase. Two things will happen. They have to align on the center of the plate, then they have to attach the mitotic spindle. Then only the cell will move from metaphase to anaphase. If not, again in the metaphase only the cell will be arrested. Then once it is arrested, again it will go through the various process to align all the duplicated chromosome in the center. Then cell go to the next phase, that is anaphase. So these are the different checkpoints there in the cell cycle to ensure that whatever activities are supposed to be done in the various phases, they see that those activities are finished, and then only the cell enters from one phase to other phase. I'm saying again, there are 5 cell checkpoints, one at the beginning of the G1 checkpoint, and the second checkpoint at the transition between the G1 to S. That's also called as a restriction point checkpoint. And there is a checkpoint in the S phase, S phase checkpoint, and the G2 checkpoint, and in the M phase checkpoint, right? So this is all about the cell checkpoints. So you can see the stop. You see stop. See one stop in the G1 at the early stage of the G1, and one more stop at the transition between the G1 to S, and one more stop in the GOS, and one more stop in the G2, and one more stop in the M phase, that is a M spindle assembly checkpoint. So there are 5 checkpoints which ensures the every activity corresponds to the every phase is done or not, right? So this is a completely coordinated activity, and each phase have a different activity, but those different activities has to be completed to enter into the next phase. So the next phase is all depends on the progression of cell from one phase to other, is all depends on the assigned or corresponding activities of the particular phase has to be finished. Then only those cells are entertained to move from one phase to other phase. If not, those cells are arrested. The arrested means delay in the progression. If the damage is not repairable, the same cell cycle inhibitors, which are inhibiting the CDK, cyclin-dependent kinases and cyclin complexes, they initiates the apoptosis by inducing the pro-apoptotic factors. So this is all about the cell cycle checkpoints. Thank you for listening. Here is my YouTube channel name. That's Dr. K PREM-PRIMER. If at all you don't want to miss any content from this channel, try to subscribe. And if you want to share, no issues. If you like it, try to give thumbs up. And if you have any issues related to the content and or any suggestions, write to me in the comment section. I'll get back to you once I see that. Thank you for listening. See you again. Until then, bye-bye.